Jonathan Challis is a research scientist with Agriculture and Agri-Food Canada at the Lethbridge Research and Development Centre. He's joining us today for a discussion on a mixture of some fundamental theory around studying the fate and behavior of pesticides in the environment, mixed with his actual research related to these topics.
So yeah, thank you for the invitation to talk today. This is my first time, I guess, teaching a course for this pesticide applicator course. So if there's any feedback, let me know. I hope this is of use to some of you. And I also don't mind being interrupted. I don't know how usually these go, but if you have a question you're not understanding, I don't mind being interrupted, but I'm also happy to take questions at the end. So yeah, I'm going to talk today about the fate and occurrence of pesticides in the environment. This is my research area of research at the Lethbridge Research and Development Centre. I've been here for about 3 years now, and that's sort of what I've been developing my research program around is trying to understand the occurrence of agriculturally related chemicals in the environment, and what are the potential impacts those may have on our ecosystems. I do study more than just pesticides, but pesticides are a large part of my program. And given the relevance to this course, that's what I'll be focusing on. Just about myself a little bit. I'm from Manitoba originally. I did my graduate schooling there, and then I did a postdoc at the University of Saskatchewan. And I moved out to Lethbridge at the beginning of 2022 to start this position. So I just want to start off a little bit talking about, I guess, toxicology, because in my world of environmental chemistry, I'm generating a lot of data to basically explain and understand what the occurrence of chemicals are in the environment or pesticides are in the environment, what their concentrations are at, where they're coming from, what they're doing in the environment. But a lot of the questions I get at the end of that data are like, well, what are the implications and what are the effects in the end? And so I work with a lot of environmental toxicologists. I do a little bit of toxicology myself as well, but it's not my background. And I also do some risk assessment. So I guess I want to demonstrate where sort of my piece fits into the puzzle. Ultimately, I'm trying to determine the risk of these chemicals to the environment. And the risk is made up of two things: toxicity and exposure, right? If you have a very toxic chemical, but there is zero exposure to yourself, that poses no risk to you, and vice versa, right? So those two things have to come together to pose a risk to the environment. And so what I do around environmental chemistry and environmental measurements is I sort of try to understand the exposure piece of this puzzle. And trying to better understand the environmental chemistry is important to define what the exposure is, right? So trying to understand where a chemical goes once it's released into the environment. Is it persistent? Does it break down? Is it bioavailable to organisms? Can it be taken up to organisms? All of those kind of questions are things that I try to address to better understand what the potential exposure could be. And then I also study and research and develop better and new ways to sample and measure these types of chemicals, because the tools we choose to use to measure and sample these chemicals and these environments can influence the conclusions around the exposure. So I sort of do both of these things in my research. And that's sort of the goal, I guess, of this little course that talk about for the next hour. Basically, I want to provide sort of a high-level overview of environmental chemistry and how we study it and some of the core principles we use to define chemical fate, and then also talk a little bit about the tools and approaches we use to sample pesticides in the environment. So I'll start with environmental chemistry. Also, just quick question: do I use the whole hour? Should I go right to 10:50 or?
If you want to leave a little bit for Q&A.
Okay, okay, cool.
Oh, thanks for checking in.
Okay, so environmental chemistry, this is sort of the Bible for environmental organic chemists. If you ever take a course in it, it's sometimes the textbook they use. And this is not actually a great figure, but I sort of use it to demonstrate the complexity of trying to understand environmental chemistry. So this is a very simplified environment where you have water, sediment, atmosphere, right? And all of these little arrows and everything are different processes that a chemical can undergo once it enters the environment, right? So, it can go from the water phase and it can attach itself to particulate matter and sediment and fall down to the sediments, right? It can infiltrate into groundwater. It could volatilize into the atmosphere and enter the atmosphere, or it could be deposited through rainfall back into the aquatic system. There is breakdown by sunlight. It can degrade the chemical. It can interact with microbial species and break down. So there's just a myriad of different processes and reactions that can occur that have the potential to move and change these chemicals, which make it challenging to study them and understand what's happening to them. So just a very general, my sort of description of what environmental chemistry is the description and quantification of the physical, chemical, and biological processes that a chemical subjected to in the environment. And I break these down into sort of two major processes, transport and movement of these chemicals and transformation of these chemicals. So one is simply moving these chemicals around into different environmental media. The other one is actually changing or altering this chemical into a new and different chemical. And those are called transformation products usually. So just another way to look at this, right? We have transport mechanisms in the blue, and these don't change the chemical. This is still your parent pesticide, whatever pesticide you want to consider, atrazine or imethoxam, unchanged, and it's simply moving to different parts of the environment, right? Transport, mixing into different compartments. You can have air-water exchange, sedimentation, things like that. Examples of these are processes like volatilization, so evaporation into the atmosphere. Absorption is an important one when we talk about pesticides, given that a lot of these things are applied onto agricultural fields, they're going to bind onto the soils. The degree to which they absorb to soils limits their movement, right? So if they're absorbing more tightly to soils, they might not be as readily moved off of those fields into aquatic systems during a runoff event, for example. Some examples in the orange here are transformation processes or transformation of a chemical. This is actually chemically changing your parent chemical to something different, right? So bonds are actually breaking in the chemical and you have something new at the end. There's biotic and abiotic processes. So biotic, an example of that is microbial degradation, right? So lots of microbes in soils, in water, and sediment. They're always looking for carbon sources, energy, and the chemicals, pesticides, have lots of carbon molecules in them. They can use those molecules to break down and use those as energy, so they'll break them down. Examples of abiotic processes, so basically chemical reactions. Photolysis is breakdown by sunlight, which I'll talk about. Hydrolysis is simply the reaction with water. So I'm just naming a few here, but these are some examples of what can happen to these chemicals in the environment. Okay, forgot I had these slides. I've sort of just defined these already, but just some of the definitions of a few of these transport processes. I guess bioaccumulation, I didn't mention. This one's a little bit weird. It technically is a transport type mechanism because it's simply the idea that a chemical in your water, for example, can be taken up into an aquatic organism or in the soil into a terrestrial organism. That isn't necessarily changing the chemical. That said, typically these organisms have the ability to metabolize and break these down. So generally speaking, a lot of these transport processes go hand in hand with transformation reactions as well. Again, these are some of the transformation processes. And at the end of these sort of reactions, what you get is what we call transformation product. So the parent molecule is no longer there and you have a different molecule. may have different effects, different behaviors in the environment. So I'm going to sort of dive into one of these fate mechanisms called photolysis, partly because I've done quite a bit of research on it. And it's, I don't know, I'm just biased. I like the idea of photolysis, which is basically the breakdown by sunlight. So it's a very common and important mechanism to understand. It can happen on terrestrial environments if, for example, these pesticides are on plant matter that are exposed to sun. Obviously, if the pesticide enters a soil environment, there is a lot less light entering a soil environment. So photolysis is maybe a bit less relevant, but in aquatic systems, hugely important to degrade these chemicals. So in general, you have sunlight and you have two different types of photolysis. Direct photolysis is the idea that photons from the sun are reaching the Earth's surface. Those can enter a water column, and that energy can be absorbed by our pesticide. And that energy is sometimes sufficient enough to actually break bonds, and that chemical will break apart. And that is photolysis, or breakdown by sunlight. And so it's just the direct absorption by your pesticide or chemical. Indirect photolysis is a bit more complex. I'm not going to talk about it too much, but I'll mention it. This is the idea that there are millions of different species in our water, nutrients, right? Organic matter, nitrates, phosphates, carbonates, species. All of those have the ability to also absorb light and absorb that energy. Indirect photolysis is the idea that some of those other species can absorb energy and then transfer that to a pesticide and break it down. So it's just a more indirect process, but still originating from energy from your sun. One more sort of thing I'll mention with just direct photolysis is that an important consideration, this OM stands for organic matter. In many water systems, you have varying degrees of organic matter, and often that leads to different colors of your water, literally. That's why I'm showing this picture up here. Typically speaking, a really brownish water is going to have a lot of organic matter and detritus and material like that. You might have turbid waters. You can imagine that in a browner water like that, you have less light penetration into the water column, less opportunity for sunlight to enter and photolysis to happen. So there's sort of like an attenuating effect there, depending on the water system you're studying. So just a couple of definitions of terms that I'm going to briefly talk about. Direct photolysis, again, is this transformation due to absorption of light, photons, direct absorption, right to the chemical. Indirect, this is the idea that it can be absorbed by other species in the water and then transferred to your chemical. And then quantum yield is one other characteristic term that we often measure. And this is the idea that it's a characteristic property that if you measure it, it tells you how efficiently a compound will break down when it absorbs light. It's a really important property to try to, if we want to try to sort of predict what a compound will do when it's exposed to light. And it's quite complex to do these experiments to measure them, but the general definition is fairly simple. It's simply a ratio of the number of molecules that break down over the total number of photons that are absorbed by that molecule. So it's simply a ratio between 0 and 1. So just for an example, a quantum yield of 0.02 means that 2% of the light photons absorbed by a chemical will result in a phototransformation. So you can imagine the higher quantum yield, if you have a quantum yield of 0.2, that would mean 20% of the time it absorbs light, it's going to break down. So a higher quantum yield means more efficient degradation by sunlight. This is an absorption spectra. These are wavelengths on the bottom here, and absorbance. And this is an important parameter to consider when we're talking about photolysis. The yellow line here is a very crude outline of what sunlight output is as it reaches the Earth's surface, right? Luckily, our ozone filters out the more intense UV wavelengths, right? And so essentially what's relevant at the Earth's surface is anything greater than 300 nanometers. So this is just important to show that basically, if we understand the absorption of our different chemicals, so this is, for example, thiamethoxam and clothianidine, two important neonicotinoid insecticides, you can see that they both overlap to differing degrees with the output of sunlight. If they overlap with the sunlight, that means that they are going to absorb light from the sunlight. And so therefore, we can predict that they probably will undergo photolysis. The other thing I'm just showing here in the orange is natural water, just to show you that natural water has species and organic matter and things like that that also absorb light. And that speaks to sort of the screening of light and the attenuating factor that it might have in a water column. So what do these experiments look like? They look, depending on what you want to do, they look very different. So we've done all sorts of experiments, but one really common thing is to study the breakdown of these compounds in the lab using what's called a photoreactor. The idea is that you very controlled in the lab, but it's meant to simulate output of sunlight. Depending on the photoreactor, they some do better jobs than others. It's actually simulating the sunlight, but still useful for studying these mechanisms. In many cases, you want to actually use natural sunlight. So this is an experiment we did where we try to, still use controlled test tubes, but are actually using natural sunlight to be more realistic. And then moving to a system that is very close to a real actual aquatic system. For example, these are called mesocosms. They're 3,000-liter tubs that have basically just miniature ecosystems in them: soil, plants, water. They're basically natural water. And so we can do controlled experiments in these, but have them totally exposed under natural systems, under natural conditions. Okay, some actual data. I won't try not to get too much into sort of the specifics of all of this data because it gets a bit complex, but essentially this is concentration on the y-axis and time on the x-axis. The way we do these experiments is we irradiate or expose these chemicals to sunlight or simulated sunlight, and we take samples over time. And then we measure them and watch how these chemicals degrade. The main upshot here is basically the steeper the line, the quicker the degradation. The shallower the line or shallower the slope, the slower the degradation, okay? This is, again, for thiamethoxam and clothianidine, and these are degrade quite quickly. This is fairly quick degradation. We're talking on the order of minutes to around 2 hours. That's a half-life I'm talking about. So this term T half means just simply the time it takes for 50% of the original concentration to degrade, okay? One thing I guess I want to sort of mention here and point out is that we study these as a function of water pH. So you go to different aquatic systems, they have different pHs. Some are more acidic, some are basic. We wanted to understand how that affects photolysis. And as you can see, it clearly does, right? For thiamethoxam, in the gray here at pH 11, so much more basic waters, you have by far the quickest degradation. Whereas for clothianidine, it's actually the complete opposite. At low pH, at pH 4 in the blue, you have much more rapid degradation. So this is just to highlight the idea that it becomes really, really challenging to understand the fate and photolysis of these things because you can't just study these under one set of conditions and assume that those parameters apply to any environment that you're going to study, right? And that is one of the challenges, is it's really hard to predict the fate of these things. We do actually have to do a lot of the measurements in many cases. So I'm just showing this one more time, just because the next set of data is going to expand this list of compounds to a few more neonicotinoids that we added into our method: imidacloprid, acetamiprid, and thiacloprid, all neonicotinoid products. But you can see that they have varying degrees of overlap with that sunlight. So we might expect that some of these compounds that barely overlap with sunlight output past that sort of 290 nanometer cutoff might not degrade as quickly as the other ones. So same type of plots, concentration over time. That is in fact what we see. So methoxam, clothianidine, and imidacloprid on this graph on the left degrade quickly. These are these three down here. We're talking about 10 to 20-minute half-life, so very, very rapid. Whereas thiacloprid and acetamiprid on the right here, quite a bit slower. We're talking about 26 hours to 42 hours, so 1 to 2 days basically, half-lives. And then I had mentioned the quantum yield earlier. I'll just show you that data because we also measure those. Remember, the larger the quantum yield, the more efficiently it's going to degrade by light. And that's in fact exactly what we see. So these three compounds that are degrading very quickly have higher quantum yields. These are quantum yields here for each compound, right? Whereas these two compounds, thiacloprid and acetamiprid, have much lower quantum yields. So they're less efficiently breaking down by sunlight. They're basically more resistant to photolysis. Okay, so moving to sort of a different question around the effect of light attenuation through a water column. All of these experiments were done in test tubes. And as you can imagine, those do not replicate a real surface water, right? Where, yes, it might replicate the very surface of the water, but as soon as you go down in depth, right? And especially depending on the type of water system, you're going to get a lot of filtering out of light going through. So, it might be probably common sense a little bit to think that, right, as you go deeper down in a water column, less photolysis is going to happen. And that was pretty obvious today, but to us, but we wanted to quantitatively actually determine the effect of that because that hadn't really been done. So we set up these experiments where we had these racks where we were able to basically deploy these test tubes at different depths in an actual water, right? Using these simulated little mini ecosystem tubs. So what do we see? We saw basically what we expected, but I guess maybe the extent of reduction in photolysis did surprise us a little bit. So again, these same plots, concentration here on the y-axis and time, now in days, right? These are exposed to natural sunlight, exposed to our photoreactor, so slower degradation under natural sunlight. But again, at the surface, so 0 centimeters, those tubes, pretty rapid degradation, right? But as soon as we went down in depth, so 8 centimeters, 18 centimeters, and 28 centimeters, we saw rapid reduction. So these are those lines up here, right? Very, very shallow slope, like change in the line. And in fact, at 28 centimeters, we detected 0 change over the 2-week experiment. So we couldn't even detect a change in concentration over time. And it basically, to put numbers on that, it was at 8 and 18 centimeters, photolysis was reduced by 90 and 98%, respectively. So pretty significant effects. And so this was just really sort of important to understand because it helped us explain the persistence of some of these neonicotinoids that we were seeing in surface waters, right? So despite a lot of lab data saying, well, these should degrade very, very rapidly in the order of minutes or a day or so, we weren't sort of seeing that in the environment. What we're seeing in the environment was we were always measuring these things. And yes, of course, there's constant inputs of these too, but it didn't jive with what we were seeing from the lab. And so what we think is that basically the explanation is that there's a lot less photolysis going on in natural systems than we think based on our lab studies, right? Because of light attenuation that's occurring at depth in these aquatic systems. Okay, so I mentioned transformation products. This is also an important consideration, right? I'm talking about these half-lives where, in 20 minutes, 50% of this compound is gone, the methoxam is gone. Well, it's not gone to nothing, right? It's just changed. So my methods measure this specific parent molecule, the thimethoxam with clothianidine. we don't have methods to measure these transformation products, partly because we don't know what the transformation products are. So we use very sort of specialized instrumentation to screen for the production of some of these other transformation products that might be formed. And so that's what we did with this work. After the experiments are done, after the 1 or 2 day experiments where we irradiate these chemicals, we then put these through an instrument that can try to pick out what are the products that were formed from this original compound. And we so established some novel transformation products that were formed. You can see that some of these look, similar, but certainly different from the parent dimethoxam. Some look very closely related, right? So a transformation product can simply be one carbon molecule just being lost off the chemical, or an oxygen leaving the molecule. That's a transformation product. It might look almost identical, but it could have very different fate properties and toxicity. So we always have to be asking ourselves, what are these degrading into? And are they posing less of a risk than the parent molecule? Often the answer to that is yes. They are usually less stable and less toxic, but there certainly are exceptions where we've found transformation products that are actually potentially more potent and maybe more stable. So it's something that we always constantly have to be at least considering. Okay, so just to wrap up this photolysis part, in general, these class of neonicotinoid insecticides degrade relatively quickly by photolysis, which is a good thing. The pH and certainly a lot of other conditions, we just talked about pH today, but pH and in general water quality parameters have a great impact on photolysis, so they can. So that's an important thing to consider. And then we also showed that sort of lab test tube data really only simulates like photolysis that's occurring right at the surface of the water and doesn't account for any attenuation that's happening at depth in a real surface water. So that's another important consideration. I was told I have to give some multiple choice questions. So how does this work?
So you have that first one up, so I'll launch it.
Okay, nice.
So the poll is the same that's on the screen. If you do have any challenges, do feel free to put your answer into the chat and it will count just as well. It looks like almost everybody's gotten an answer in. Just 80% now. Seems like this poll worked better. All right, we'll give it a couple more seconds here for folks to get their answers in. And again, if the poll is not working for you or not popping up, feel free just to put your answer in through chat. Seems like almost everybody's gotten the opportunity to answer this poll. Feel free to move over to the next one, Jonathan.
Okay, so I'm going to switch gears a little bit and talk about still environmental fate, but now just simply a transport mechanism, not a transformation mechanism. And I'm going to talk about long-range transport or atmospheric transport. I chose this just because I find this whole idea pretty fascinating. It's sort of actually what got me interested in environmental chemistry back in my undergrad. But it's the idea that there are these semi-volatile organic compounds, many of which are pesticides. And when I say semi-volatile, volatile means a highly volatile compound will want to just evaporate into the atmosphere and stay there. And there's lots of examples of highly volatile chemicals. Semi-volatile compounds are sort of in between. Under the right conditions, they will evaporate into the atmosphere. Under other conditions, they will be deposited and persist in the water. They will bind to sediments. So there's sort of these compounds that exist in between a lot of different environmental compartments. And what was being found when in sort of throughout the like mid 1950s and on, we started measuring, I guess a little bit later than that. The use of these chemicals would have been in the mid 1900s and even earlier. And that's when they would have been starting to transport to the environment. But we were finding, environmental chemists started measuring chemicals that were in the Arctic, in the poles, basically, North and South Pole, in Arctic ice, in seawater, in organisms like beluga whales, seals, right, that accumulate these things, which was a bit of a mystery because obviously these types of chemicals, like industrial chemicals and agricultural pesticides, are not used anywhere near the poles, right? And so basically what was theorized is what's called this grasshopper effect or global distillation. And it's this idea that these semi-volatile compounds can be used and generated and emitted sort of at lower latitudes where there's, generally speaking, higher populations around the globe and higher industries and agricultural practices going on. And so they're used at the lower latitudes, but they are semi-volatile. So under the right conditions, i.e. in hotter temperatures, they will evaporate into the atmosphere and be transported sort of north or south to higher latitudes. But there's seasonal cycling. So they're semi-volatile. So the volatility really depends on the conditions. So they're not just going to evaporate and stay there. Basically, they will evaporate and then move to colder, higher latitudes, colder temperatures. Winter season comes along. Cold temperatures don't favor volatilization, they favor deposition. And those chemicals will be deposited back down to a terrestrial environment where they will stay until, say, the next season where summer comes along. You have higher temperatures and it will start evaporating again and being transported to higher and higher latitudes. And each sort of cycle of this is like sort of as a hop, right? That's what this is showing. This is just imagining a grasshopper hopping along the latitudes. What happens eventually is that it reaches the poles. And as we know in North and South Pole, temperatures are extremely cold and essentially never get warm, right? That doesn't favor evaporation, that favors deposition. So what happens is that these chemicals eventually make it to the poles and they become trapped there. It's called cold condensation. They get deposited and stay there essentially forever. These chemicals are extremely persistent. They essentially don't break down. And that's how sort of how we came to understand the transport, long-range transport of these semi-volatile compounds that in theory shouldn't be transferred that long range based on just their volatility. And so just to give you some examples of what these are, first of all, highly volatile compounds. I'm not talking about those. Those are compounds like, refrigerants that are highly volatile. They get evaporated and just stay there. Those can be transported. Those are transported all over the world consistently. But these semi-volatile compounds are more things like legacy pesticides that are highly toxic and just really nasty compounds. Things like lindane and heptachlor and dieldrin and DDT. Many of you have probably heard of DDT, certainly banned here, but still actually used in some parts of the world to control malaria. Not because it's not a toxic chemical, but because the risks of malaria outweigh the risks of DDT exposure, essentially. But all of these compounds are still, despite their being banned almost 20 years ago or more across the world, they are still measured in the Arctic today and will continue to be. And that's because they're trapped there and they really have no mechanisms that are able to degrade them naturally. A lot of these legacy pesticides were part of the Stockholm Convention, which was a convention that looked to ban and still does environmental contaminants that are problematic for the environment. And many of these legacy pesticides were part of this very initial signing of the Stockholm Convention called the Dirty Dozen. These 12 highly toxic and persistent POPs or persistent organic pollutants, right? There are, and part of that 12 are also things like polychlorinated biphenyls, PCBs, which are industrial compounds used in electronics, dioxins and dibenzofurans, basically some of the most toxic chemicals humans have ever produced, essentially. And a lot of them look like this. They're these carbon rings with chlorines on them. Fortunately, we've got a lot better at designing safer and better pesticides and chemicals in general that don't have such a broad, toxic profile, and they don't persist in the environment. So the idea is like design a chemical that has a targeted effect to what we want it to do, but ensure that once it's done that job, it's not so persistent and it doesn't have non-target effects. A little bit more of just sort of like a more slightly relevant and recent example, chlorpyrifos, which was a common organophosphate. It has been banned in Canada since 2021, I think. But that's another example that of a pesticide, more current use that underwent long-range transport. So chlorpyrifos has and still is in some cases measured in high latitudes where it's never been used. So that's atmospheric transport. I don't have any, I'm not going to show any sort of specific data about that, but I wanted to introduce that idea. So I'll sort of just conclude this part of my talk on environmental chemistry. Just to say that I think hopefully I've demonstrated that studying the environmental fate of pesticides can help us understand where these chemicals are going to end up once they're in the environment and how they will or will not degrade. And that information is critical to understanding the potential exposure of pesticides to non-target organisms. Non-target being, things like fish in an aquatic environment or amphibians or, other terrestrial organisms.
There's a question from Wade Dunham in chat. Does hydrolysis breakdown still happen at the poles?
Yeah, for sure. It would break down. Absolutely. Yeah. Anything in water that's exposed to water, if that's a mechanism that can break it down. So it all has to do with the chemistry of the molecule. And hydrolysis is very dependent on pH also. So for example, like more acidic waters, you get what's called acid-based or acid-driven hydrolysis. So it depends on the conditions of the water. But just as a note, the chemicals that some of those legacy compounds that I showed that are showing up in the poles, those do not undergo hydrolysis at all. There is no mechanism where they react with water. So part of the reason that they're so persistent. So yeah, and then just to hammer home this other point, right? That understanding exposure is critical to assessing risk and doing accurate risk assessment. And I guess just one more final point, like this study of environmental fate is like not just an academic exercise. A lot of these parameters that we define around environmental fate are actually used in policy and in some cases law. And so just for an example, this is right from the Canadian Environmental Protection Act, where we define basically half-lives of compounds in different media. And that sets a sort of a strict definition of whether or not a chemical is persistent and can undergo long-range transport, for example. So just highlighted in the box above here, in air, a half-life of a chemical equal to or greater than 2 days is considered persistent and can undergo long-range transport. So you might not think 2 days is very long, but that's a product of the fact that a chemical in the atmosphere can be transferred such long distances in very short periods of time, right? So it doesn't need a very long half-life to be transported around the world, essentially, through atmosphere. In water, obviously that looks very different, right? Transport in water is going to be a lot slower compared to the atmosphere. So what we consider a persistent chemical in water is a half-life greater than or equal to 182 days. So very, very different considerations, right? I didn't really talk about bioaccumulation, but that also shows up in the Protection Act, right? Defining whether or not a chemical has the potential to bioaccumulate into organisms. The idea that they will accumulate chemicals and then they will stay there and sometimes concentrate those chemicals until their bodies have pretty high burdens of these chemicals. So that's actually, you can calculate a bioaccumulation factor. And so anything over 5,000 is considered to have bioaccumulative properties in chemicals. So just before I switch gears to like environmental sampling and some of my other data, I want to talk a little bit about environmental risk assessment because it is something I do, and it's sort of the end product of a lot of the data that we generate. And I want to talk about the process of doing environmental risk assessment and how we sort of establish protective guidelines for individual compounds. And the best way to do that is by using what's called species sensitivity distributions. So this uses the toxicity of a chemical to given organisms, and basically it develops a distribution of toxicity and sensitivity. So the way this works is here you have the percent of species potentially affected by a single chemical now. Each that species sensitivity distribution is developed for an individual chemical, and then you have a concentration of that chemical on the bottom here. And so what you do is you go to the literature and say we're studying a lake. We're going to try to pull toxicity data for all relevant species in that lake: fish, aquatic insects, aquatic plants, amphibians. We want to get the broadest possible distribution of species in that lake to be representative, okay? And then we take those toxicity data that have been studied for these compounds. So that could be, for example, an LC50, that means a lethal concentration that kills 50% of the population, or an EC50, an effective concentration, that just means a non-lethal endpoint. So say you're looking at the effect on reproduction or the effect on growth or some behavioral endpoint, right? So the concentration is going to have a 50% effect on that population. And then, so those are then plotted on this graph. And so what this looks like is species up at the higher end of the distribution are less sensitive. They require more chemical to elicit the effect that's being studied. Whereas species on the lower end of the distribution are much more sensitive. You require a much lower concentration to elicit that same effect. And then what's standard for basically developing protective guidelines is looking at this 5% value. So you take the 5% and just extrapolate down to the concentration. And that's what's referred to as an HC5, or a hazardous concentration affecting basically the 5th centile of species. And that's used in the generation of protective guidelines. And so just to show you what that looks like in reality, this is an actual species sensitivity distribution. I don't know what these species are. This is the actual species name, but they're grouped by, amphibian, fish, and vertebrate plant. You can see that you have a very wide range of different species, which is good. And so we look at basically this 5 percentile value to be very protective. And what that means is basically that 95% of the species in the system will be not affected by that concentration. 5% will. Okay, and so then to develop a water quality guideline, we apply what's called a protection factor or an uncertainty factor. Some people call it a safety factor. These values, sort of 10, 100, 1,000, they vary depending on who's doing the risk assessment. So depending on the government agency or the group or the perceived risk of a chemical, you may use a more conservative or less conservative safety factor. For example, Europe tends to be much, much more conservative on these things. So the European district or agencies generally use higher uncertainty factors. And so the way to generate a water quality guideline then is to take that HC5 value and divide it by your uncertainty factor of, say, for example, 100, right? So dividing by 100 now brings that value way down here, right? So it becomes very protective. And the reason for that is that you have to consider that, for example, it's impossible to capture all species in a given system in a species sensitivity distribution. So maybe maybe the off chance this one or two species that you didn't include in your species sensitivity distribution happen to be the most sensitive to that chemical, right? So it's sort of accounting for some of that uncertainty. There's also uncertainty around the individual toxic endpoints and those values from the literature. So it's accounting for that. So it's just the idea that you want to develop guidelines that are you can safely say are protective of an environment. So that's the upshot of sort of environmental risk assessment. So what we can end up doing now is what I do with my data in terms of the exposure data is I can take all of the concentrations that I measure in the environment and start comparing them to these guideline values and trying to say, are they exceeding these values? Are they posing a risk to the system? Okay. I didn't know I had this already. Okay, another multiple choice. This is the last one. Just
Should be
around
live now.
the grasshopper effect. And then I'll switch gears a bit.
Answers are coming in quite fast. Already 3/4 of the audience. Give it a few more seconds here. In the meantime, Darren Van Ray put a question in chat. Does the snowpack in the Arctic absorb chemicals as well?
Yeah, absolutely. Yeah, they get deposited into the snowpack. That then gets compacted down and forms ice in many cases. And a lot of the research actually is really interesting. What you can do in the Arctic is you take snowpack cores or ice cores and you date them, and then you can start looking at like cutting those cores into pieces, dating them, and determining when actually individual chemicals were deposited in the Arctic or Antarctic or wherever you're studying. One really interesting sort of application of that was because these also accumulate in Arctic, a lot of Arctic species, polar bears, and then whales and seals and all sorts of things. Unfortunately, they're very susceptible because those species, especially like those aquatic species like seals and beluga whales, have a lot of fatty tissue. And these chemicals love to partition into that fatty tissue. And so what happens is they basically just are like sponges for these chemicals. And then what happens, it's in their fatty tissue, which doesn't necessarily have an effect on them immediately. But then as they are migrating and maybe not eating them as much, they're all of a sudden burning a lot of calories, losing a lot of that fat. And all of a sudden those chemicals get remobilized into their system. And that's when they can actually have the toxic effect. But yeah, also one really interesting example I just mentioned of dating accumulation of chemicals was some research in Arctic on whales where they were able to take cores from the earwax of a whale and cut them up and date them and observe like which chemicals were there and when they were deposited. Really funny example. They had a picture of the actual earwax core and it's like enormous.
Interesting. The poll is over now, so feel free to continue.
Okay. Time we got. So I've got, okay, I don't know how much I'll get through here. That's okay though. Yeah, I'm switching gears to talk about a little bit now about the tools and approaches that we use to actually sample pesticides in the environment. And so it's really important to monitor, these chemicals from an exposure and toxicological perspective, right? In order to understand risk. And just having baseline monitoring data in these systems is really, really crucial if we want to try to understand how these systems are changing when there's an exposure to chemicals like pesticides, right? Because if we have no data before exposure occurred, we don't really understand how these systems are changing for better or for worse. So when we're doing environmental sampling, there's lots of things to consider around type of sampling approach we use. The sampling sites and the selection of them are critical. Where, when, frequency, the number of sites, control sites, the cost of the program. We need to ensure that the sites we're choosing are going to answer the questions that we're asking, right? So are we trying to determine the source of the chemical? Are we just trying to identify what the peak concentration is that's occurring, or do we want to know more of what's an average concentration? Is it going to be a long-term monitoring program, short-term? These are all sort of important questions to ask before trying to design a program to sample pesticides in the environment. And so we'll run through a few techniques that we use in our lab. Active auto samplers are this idea that you deploy these systems that require a power source, and their value is that they can automatically take water samples based on certain intervals or triggers. For example, as the water level changes, you can trigger it to take a sampling event or just time intervals. Grab sampling, as you can imagine, very simple use, going to a system, dunking a bottle of water in and bring it back to the lab. And that is very valuable because it's cheap and easy to do, but keeping in mind that it provides a single snapshot of the system in time, right? Passive sampling is maybe something you haven't heard of, which I do a lot of. I develop passive sampling tools. What these are these non-mechanical devices that get deployed in an environment for extended periods of time. And as water and chemical passes through these devices, there's basically you can think of it as a sponge that has a high affinity for pesticides or the chemicals we're interested in, and they will be pulled from the water onto our sampler. And so we can sample basically continuously during the deployment period. And so these are cheap, they're easy. It's I like to think of it as a very happy medium between grab sampling and actual active auto sampler, because these do not require any moving or powered parts. You can also have passive air sampling in the air, same idea, but you're deploying them in the air. You can have active samplers for air samples. We actually have these units in our lab. We do some air sampling where there's a pump that actually brings and filters air through. And yeah, just a couple more pictures. These come in lots of different shapes and sizes, and they're designed for different things. You have water samplers, air samplers. This is just a schematic, but this is actually a soil probe. So you put it into probe, and you can have passive samplers for sediments or soils, things like that. So there's all sorts of different things you can do. And I guess with this figure, what I want to demonstrate is the type of information we can get from a grab sampling versus a passive sampler. So you can imagine concentration of chemical, pesticide, whatever you want to think about in a system is going to change over time, right? You might have a heavy rainfall event and a pesticide gets flushed into an aquatic system, or you might have a pesticide application. And that might, drift off spray and end up in the aquatic system. So over 1 month, you're going to have changes in concentration. A passive sampler can be deployed, for example, for 3 months. 3 weeks. It's continuously sort of sampling and integrating over these peaks and valleys in concentration. We end up getting is this dashed line, which is sort of basically an average concentration of the system. What's useful about this is that it's typically more representative of what an organism is exposed to in these systems when we're trying to determine risk, because yes, of course, an organism is exposed to a peak concentration, but that quickly attenuates and goes down back to sort of baseline levels. And when an aquatic organism is swimming around there for its entire life, it's going to be somewhere, in general, on the aggregate, exposed to sort of an average concentration. Whereas for grab sampling, it's very simple, but it's challenging to do frequently, right? Especially if you've got one of our programs samples 70 different sites across southern Alberta. it's literally impossible to do that more than once a month. So that's what I'm showing here. Here's an example of a grab sample. It's an instantaneous snapshot in time. We don't know what we're sort of getting in the system. Are we getting a very low point in that system of that chemical? Are we capturing a peak? We have no way of knowing when we just take grab sampling really infrequently. And so that's sort of the value of passive sampler, is that you have more ability to capture events that flush chemicals into an aquatic system. But in general, these two sort of are complementary, and that's how we use them as well. And I'll just sort of mention sort of the history of passive sampling, I guess. These originated as organisms. Organisms were the original passive sampler. This is called biomonitoring, and it's still done today. But it's the idea that organisms will accumulate, like I talked about in the Arctic, for example, will accumulate these chemicals in the environment, and then we can sample those and determine exposure directly, right? And so going back to the 1800s, lichen has been used to assess air contamination. And so there's some criteria to define a good sort of biomonitoring organism, but there's all sorts of different ones you can use. And so basically passive sampling, the idea is like this same idea, but removing the biological part of it, because it's very annoying to deal with fish species or mussels or living organisms. It's a lot easier to deal with this non-mechanical device that can do the same thing. I think I'm running out of time here. So I'll just quickly show a little bit of data that I have from our work in the irrigation districts. We measure pesticides in irrigation water. Our lab has been doing this for a long time, predating me. And I've recently sort of secured some new funding to add to the methods and study different sampling techniques and things like that to enhance this monitoring program. But the irrigation districts sample 70 sites over the growing season, and then we get those samples and we measure them for over 100 different pesticides. So here's what that data looks like. Each of these peaks represents an individual pesticide, and then we are able to relate basically the peak area. The greater the peak area, the greater the concentration, and we can quantitatively relate that to actual concentration in the water. And I'm going to skip because I'm running out of time, but what I just quickly want to show is exposure distributions and how we can use these. So what these are all of the concentrations that we measured in these irrigation districts for a given compound. Imidacloprid, atrazine, methotrexate, tebuconazole. These are just 4 examples. Again, we measured 100 pesticides or over. And what we're doing this is plotting these concentrations as a function of percent occurrence or probability. So the way to look at this is that at the top of this distribution, you have the highest concentrations. At the lower end, you have the lowest concentrations. And what you can do, and I just wanted to mention this because it speaks to the development of those water quality guidelines, right? We can now take guideline values like the Canadian Council for Ministry of Environment, the freshwater guideline for imidacloprid is 230 nanometers, and nanograms per liter. We can plot that on this graph and see where it crosses this exposure distribution. As you can see here, it basically doesn't cross the exposure distribution. This red dashed line is occurring to the right of this whole concentration distribution, meaning that we essentially don't ever exceed this guideline for imidacloprid, which is great. To put a number on it, is less than 1% chance of exceeding this guideline. For atrazine, it's even greater. The value is 10,000 nanograms per liter. So there's less than a 0.001% chance of exceeding this water quality guideline in the irrigation districts. So we can accumulate this data, and then we have the ability to sort of predict based on guideline values, are we going to exceed any guidelines, and is there going to be risk? And just, I guess the last comment, these are all nanogram per liter concentrations up to like sort of 100 nanograms per liter. These are very low concentrations. So, I mean, the common example is nanogram per liter is parts per trillion. So that's the equivalent of 1 drop of water in 20 Olympic pools, Olympic-sized pools. Or put another way, 1 part per trillion or nanogram per liter is 30 seconds out of 1 million years. So very, very small amounts, but our methods can measure these things. And so these are really valuable to basically pair with water quality guidelines to understand risk. Okay, it's 10:50. I think I'll just wrap up here. So yeah, I've sort of said all of these things, but I think I've shown that sort of understanding environmental fate is really critical to understand ultimately exposure and risk. The sampling tools we use can be important to understand and estimate this risk accurately. And yeah, all of this feeds into sort of risk assessment and ultimately understanding whether or not these chemicals pose risks to our ecosystems and environments. Yeah, that's it for me.
Awesome. Thank you very much, Jonathan. I'll throw the CCA QR code for this session up on the screen now. Oh, now. And yeah, any quick final questions for Jonathan before we move on? As well, feel free to quickly refill water, grab a quick snack, and then we will move into our next session with Andrea Sawatsky.
I
Great
don't think I
question.
provided my email, but also someone probably has it. Feel free if there's any questions afterwards too. I don't mind you sharing my email.
For sure, thank you. And Jamie asks, how does your work inform policy decisions?
Yeah, good question. I mean, that sort of speaks to the risk assessment part of my work and the exposure distribution. So, for example, I work very closely with the PMRA in Health Canada, they're developing a national water quality program actually to monitor pesticides across Canada, and I'm helping to generate some of that data, and I share it with the PMRA and Health Canada to inform their risk assessment on ultimately. And those risk assessments around these pesticides are what's informing registration and re-registration and policy around whether or not, the safety of these chemicals and things like that. So I don't directly work on policy, but certainly a lot of my environmental data feeds into policy and decision making around regulation of pesticides.
Awesome. And Ken Coles has a question.
Hi, Jonathan. Thanks for the talk.
Okay.
I'm just curious, most of the new products that are entering the marketplace tend to fall under the soil amendment, so they're not necessarily regulated by PMRA. So all of the regen ag products and this and that, I understand that they're supposed to prove environmental safety, but is there really much work being done assuring that we're not causing problems with the plethora of products that we're playing with on the soil these days?
I mean, I would say no, not enough evidence. I mean, I don't know about all these products, but in general, it's so hard to keep up to the production of new chemicals. And so a lot of that's why I think it's the important regulation and requirements for proper studying of these fate and behavior and toxicity of these chemicals before they reach market is really important. And so if that's, I think there's some concern if that's not being done properly beforehand, because even for a lot of these companies that are creating these pest products and providing a lot of the required data, it still falls short a lot of times of what we end up learning about these products afterwards, and the information that we need. It's not to say that, I mean, in general, I guess I'd say though, like for the most part, I think my data shows that there's good stories, good news stories around the risks of these things, but there certainly are gaps and things that get missed. And if these aren't being regulated as strictly by these agencies, I think there's potential there to for sure miss something. And then if we miss something and it's not properly regulated, it takes so long for the researchers to catch up to that, right? Because for us, we have to generate, you can't just generate one study and say, oh, look at this. This is proving something. It often takes years and years of those studies to be repeated and studied in different ways for the body of evidence to be generated to say, okay, this is actually posing a risk.
Awesome. Thank you very much, Jonathan.
Yeah,
And
thanks for the invitation. I appreciate it.
absolutely.